Spinal muscular atrophy is a motor neuron disease like ALS, Lou Gehrig's disease. However, it's also a little bit different from ALS in that it's a genetic disease, whereas ALS is more of a sporadic disease with no clear genetic association in about 90% of the patients. And also, it's a childhood disease, unlike ALS, which affects mostly older adults. It's a devastating disease, and about 70% of the kids with this disease die before they're 1 or 2. Because it's a motor neuron disease and motor neurons are very difficult to study, we were determined to try to study this disease using motor neurons derived from embryonic stem cells as an important component of our study systems. We carried out a screen that was designated to correct the problem with this disease. And the problem is that there's a protein called survival of motor neuron with slightly unknown function, but it is known to be necessary for motor neurons to survive, as the name suggests. The disease is caused when the level of this protein decreases too much. It needs to decrease to about 25% of normal, and if that happens, that actually leads to death within a year or 2. So we were looking for small molecules, drug-like molecules, that could be used to treat kids that would cause an elevation of the level of this protein. We did a very sophisticated and difficult screen which involved growing cells in culture and imaging them using an automated high-throughput screening microscope, looking for any kind of molecule that would raise the level of this protein. We found about 150 molecules that raised the level of this protein, but we focused in on a small subset of the most drug-like molecules. And one of the ones that we just described in a paper that was published in Nature Chemical Biology acts as a kinase inhibitor, and the kinase inhibitor causes the protein to be stabilized and increase in amounts over time. And when that happens, when the protein increases, it does just what we were hoping for, which is to say it keeps motor neurons alive when they would otherwise die. So the next steps in this project are to actually start testing these compounds and the other ones that we identified in the screen in mouse models of spinal muscular atrophy, and then hopefully, ultimately, we would be very excited about trying to move some of these molecules into clinical development.